Hey everyone, and welcome to this explainer. And honestly, I am absolutely thrilled you're joining me today. We are going on this completely fascinating journey into the science of exactly why we love the foods we eat. As someone who totally geeks out over food science, I can tell you that every single time you sit down for a meal, your body is doing this incredibly complex, high-speed scientific analysis. We're going to unpack exactly how that happens, from our own basic biology all the way up to the high-tech laboratories that perfect the snacks sitting in your pantry right now. But before we get into the lab stuff, let me ask you something. Have you ever noticed that a perfectly delicious frozen bowl of ice cream tastes incredible, but if you let it melt completely into a puddle, it suddenly tastes, well, almost unpleasantly sweet? Like, why does the exact same food, literally the exact same ingredients, taste completely different just because it warmed up? Well, as we'll see today, taste isn't just about a recipe. It's a deeply complex mix of temperature, biology, and chemistry. Okay, let's dive into this. Here is our roadmap. We're going to start with the science of eating, then sight, smell, and taste. From there, we'll look at texture and temperature, step inside the sensory lab, and finally check out some really cool food testing gadgets. All right, jumping into section 1, the science of eating, focusing on human sensory judgment. Okay, take a look at this incredibly intimidating-looking word, organoleptic. I know, it sounds highly technical, but it really just means evaluating the quality of a food product using our human sensory organs. So when appearance, flavor, and mouthfeel all come together to help you decide whether to accept or reject a food, you're performing an organoleptic assessment. Literally, you are the testing instrument.
So, the absolutely crucial point here is that sensory quality isn't just a fixed property sitting there on your plate. The affective characteristic is actually your reaction to it, your psychological and sensory response to the food. If you have a really pleasant memory tied to a certain dish, your physiological and psychological reaction will literally make it taste better to you. It's this super dynamic relationship between the meal and your mind.
Moving right along to section 2, sight, smell, and taste, and those all-important pre-bite expectations. Think about visual cues for a second. Sight basically dictates our acceptance before we even pick up a fork. A piece of fudge with a rich, glossy surface. Our brains immediately rate that highly. But what about the interior appearance of something? If you see lumps in your gravy or pudding, your eye instantly judges it as undesirable. No thank you. We even assess things like the lightness of a loaf of bread or the cloudiness of a glass of juice purely through our eyes. It completely sets our expectations before we even take a bite.
And then comes smell or odor. Now, for us to actually smell a food, the substance has to be volatile. That just means its molecules must vaporize and hit the receptors in our olfactory organs. And temperature plays a massive role here. High temperatures volatile those aromatic compounds. That is exactly why the rich, spicy aroma of a boiling hot pot of sambar can penetrate way beyond what you can see, basically dragging you into the kitchen from all the way across the house.
And finally, taste. Our taste buds register 4 main categories: sweet, salt, sour, and bitter. Generally speaking, the buds near the tip of your tongue are most sensitive to sweet and salt. The sides detect sour, and the back of your tongue is what warns you about bitter flavors. But here is the truly fascinating part. Everyone has a different threshold. And a threshold is just the specific concentration required for your individual taste buds to even identify a flavor. Your sensitivity levels are completely unique to you. That's why your friend might complain a dish is way too salty while you think it is seasoned to perfection.
All right, section 3, texture and temperature, and how they completely alter flavor profiles.
Remember our melted ice cream question from earlier? Well, here is the scientific answer. Human taste sensations actually become way less intense when the temperature of food drops below 20 degrees Celsius or goes above 30 degrees Celsius. Scientists call this the 20 to 30 degree rule. When ice cream is frozen solid, the extreme cold actually inhibits your taste buds, so the manufacturer literally has to pump it full of extra sugar just so you can taste the sweetness through the freezing cold. But when that ice cream melts and warms up to room temperature, your taste buds get their full sensitivity back, and suddenly, bam, you taste all that excess sugar, making it unpleasantly sweet. By the way, this is exactly why piping hot coffee doesn't taste nearly as bitter as coffee that's cooled down in your mug. And this brilliantly illustrates how texture, or mouthfeel, works hand in hand with taste.
Texture in ice cream entirely depends on the size of the ice crystals. Large crystals make it feel coarse and grainy, which we really don't want. Meanwhile, the brittleness of raw carrots offers this incredibly satisfying crunch because the plant cells are resisting the pressure of your teeth. Even the tenderness of a pastry crust is judged organoleptically by just how easily that crisp crust breaks apart in your mouth. Next up, Section 4, Inside the Sensory Lab, and the world of highly controlled environments. So how in the world do we measure something as subjective as a satisfying crunch? Well, we have to shift from our cozy dining tables to highly controlled scientific environments. A professional sensory testing lab is typically divided into very specific units to isolate all the variables. Panel members first enter a reception room just to get acquainted with the day's tasks. Meanwhile, the food is perfectly portioned out in a completely separate, pristine preparation room. Finally, the testers go into the test booths. And let me tell you, these booths are intense. They are strictly air-conditioned, entirely free from background noise, and scrubbed of absolutely any extraneous odors. It is a completely 100% neutral environment. The rules in these labs are incredibly strict, mostly to avoid human error. For instance, testing is usually done between 10 a.m. and 12 p.m. Why? Because that's when the testers' palates are at their absolute freshest. Also, to prevent any unconscious bias, samples are never actually named. They're just given randomized 3 to 5-digit codes. Get this, if scientists want to test flavor without appearance getting in the way, they will literally use colored lights in the testing booth to mask the true color of the food. No way you're getting biased by a color you can't even see. Plus, testers only get a maximum of 4 to 5 samples per session to prevent palate fatigue. Now, what really cracks me up is the huge difference between polite table manners and professional lab techniques. Like, if you're casually eating out, you might gently smell your food. But in a lab, testers take short, rapid-fire sequences of sniffs to perceive the aroma as clearly as possible. And while aggressively slurping your tea is definitely frowned upon at a nice dinner party, in a sensory lab, testers intentionally slurp a teaspoon of coffee or juice. They roll it all around to ensure the liquid completely coats every single taste bud on the tongue. So yeah, in this case, science literally requires slurping. All of those sniffs and slurps eventually get recorded on an evaluation card. But here's the catch. There is no one-size-fits-all scorecard. Designing these cards is famously difficult because every single experiment requires a custom-designed, completely unambiguous questionnaire. It has to perfectly translate a judge's highly subjective feelings into hard, actionable data for the researchers. Definitely not an easy task. And that brings us to Section 5, Food, Testing Badgets, and Heavy Machinery. So what happens when the subjective human palate just isn't precise enough? That is where the heavy machinery comes in. Food scientists have an absolutely incredible arsenal of basic and color-measuring instruments. We're talking about a digital salt meter that can tell you the exact salinity percentage of a single drop of liquid in just 3 seconds, or a butyrometer, which uses ether and alcohol to separate and measure the exact butter content in milk. And for color, human eyes are easily fooled, so they use a disc colorimeter. This thing spins colored discs at a dizzying 2,700 revolutions per minute until the colors merge into a single hue without flickering, giving them a perfect visual match. Or they might use a spectrophotometer, which actually passes a selected wavelength of light right through a liquid to measure exact clarity, cloudiness, and color intensity based purely on light absorption. Now, when it comes to texture, the gadgets get even more specialized depending on what kind of food it is. For liquids and semi-solids, scientists use a viscometer to measure rotational resistance. Think of testing the thickness of mayonnaise or custard. They also use a penetrator, which literally drops a needle into a gel or a baked good using gravity to see exactly how tender it is. But for solid foods, they bust out the compressimeter. This machine applies immense pressure to deform the sample, measuring the exact force required to do so. Obviously, the more force needed, the firmer the product.
But the absolute king of the lab is the Universal Testing Machine. This device completely destroys the food sample in the name of science. By doing so, it provides a flawless, measurable record of 7 distinct textural profiles: cohesiveness, adhesiveness, hardness, springiness, gumminess, chewiness, and fracturability. It perfectly quantifies absolutely every single aspect of what you physically feel when you chew your food. Which brings us to the end of our journey today. We've explored how your tongue maps out flavors, why a change in temperature can completely rewrite how your taste buds function, the incredibly strict rules of sensory labs, and of course, the heavy-duty machines that measure the exact chewiness of your favorite snacks. So I'll leave you with this thought. The very next time you sit down to eat, take a second. Pay real attention to the temperature, the texture, and the aroma.
How will you organoleptically evaluate your next meal? Thanks so much for exploring this with me today on This Explainer, and keep letting your curiosity guide you.
Life Science Life channel. Like, share, and subscribe.